Hello and welcome to Topic 3 in the IB Biology curriculum, which focuses on genetics. We're going to start slow and build up our knowledge of genetics in this video and subsequent videos. So without wasting any time, let's dive in. The world of genetics can be pretty complex, but understanding the basics can help you navigate this field and build a strong foundation for conceptual understanding. And in the realm of genetics, there is nothing more basic than a gene. Now, we should know that our cells have a control center called the nucleus. And within that nucleus, there are molecules that provide the instructions for how the cell operates, called DNA. The field of genetics is really the study of how DNA is used and changed over time. So back to this important term we have here called a gene. A gene is a section of DNA that gets coded to create a protein, which will determine a trait. If we look at this image, we can see that there is a very long strand of DNA, but not every section of the DNA is made up of genes. This gene, or section of DNA, that we can see here carries a specific code that can influence a trait within a cell and larger organism. All of the other sections that do not code for anything, aka are not genes, are commonly called junk DNA. Though they might not be entirely junky because scientists are discovering that they do have a purpose, like helping control gene activity. Another key part of what makes a gene is its ability to be passed down from one generation to the next via offspring. This is where the heritable term comes from. Genes are sections of DNA that code for traits that can be passed from parents to offspring. So if you happen to share some traits with your parents like hair, eye, and skin color, height, and earwax consistency, it's because they literally made copies of their genes, their sections of DNA coding for these traits, and gave them to you. So make sure to thank them for your DNA when you get a chance. If we compare two different genes, we are comparing different sections of DNA that exist on a chromosome. Now, these two genes could be on the same chromosome or on different chromosomes, but either way, they occupy their own unique spot or position. The formal name for a gene's unique spot is called a loci. Loci are named using a system that identifies that location on the chromosome, whether they are on the P and Q arm, which is the short or long arm separated by the centromere, respectively, and then by its position. The idea here is that the same gene for each person has the same loci, or same location. So for example, if you are a human, you have a gene called OCA1. If the recessive version of this gene is mutated, it can result in albinism. The location of this gene is 11q1.4-q2.1, which means it is on chromosome 11, the long Q arm, and somewhere between position 1.4 and 2.1. And this location of this gene applies to every human on the planet. It's easier to study genes if you know where they are in the genetic code, and it's easier for scientists to share information if they have the same system to share their loci. From the last slide, we know that a gene is located at a specific spot, or loci. But there has to be more to the story than just the location of a gene, because after all, we are all human. So we roughly have the same genes at the same locations in our genome. If this is true, what actually makes us look and act different with our own unique characteristics? The answer to that is alleles. An allele is a version of a gene, more specifically a unique code within a gene that creates a different version of the same trait. An example could be a gene associated with hair color. Naturally, there are a few different colors of hair that we see in our global human population. So the loci or position of this gene could be involved in coding one's hair color. But the actual sequence at this location will determine what the hair color is: brown, black, blonde, red, or an intermediate color. These different versions of the gene that determine the specific trait are the alleles. You can see with hair color we have a few options, but this is true for all of our traits. And it is a combination of different alleles that we inherit from our parents that makes us unique as individuals. So always remember, loci is the location for the trait, and the alleles are different versions of that trait at the same loci. When learning about genetics, it's important to know that changes within the genetic code can occur through a process called mutation. We know that the DNA sequence at this particular locus is going to determine a trait for an organism, and that there are different versions of traits called alleles. The reason these alleles exist is because mutations, or copying errors, can occur when the DNA is being copied in the process of cell division. We will talk about this process more in a future video, but for now, we just need to understand that when DNA copies itself, sometimes mistakes are made. Sometimes a base is added to the sequence, or subtracted from the sequence, or switched with a different base. In any case, the underlying genetic code is slightly different, which can result in a different protein being built for that trait, forming a new allele. If these alleles work well enough, they can stick around in a population and be passed to offspring, just like any other genetic code. Mutations can lead to new combinations of DNA, and while sometimes they have led to new alleles that provide neutral or beneficial outcomes, they can also be harmful. Sickle cell anemia is a blood disorder that is caused by a DNA mutation. This mutation impacts the structure of the hemoglobin protein, which in turn changes the shape of red blood cells from their regular donut shape into a half-moon sickle shape. This makes it difficult for cells to carry oxygen and move around the bloodstream, as they can often get stuck, which can further block blood flow and be very painful. This disease occurs because a single DNA base substitution within the hemoglobin gene, the 6th codon in the sequence specifically, is changed from CTC to CAC, meaning the thymine was mistakenly replaced with an adenine. When this DNA gets transcribed, it changes the messenger RNA code at that location from GAG to GUG. When translated, this changes that amino acid at that location from glutamic acid, GLU, to valine, VAL. This small change in the protein has the profound effect of altering the hemoglobin to be built incorrectly, causing the problems we already mentioned. And this gene and mutation is also heritable. This goes to show how powerful your DNA is, as one very minor change can have a large effect on the overall health of an organism. So far, we have talked about genes and alleles on an individual basis, but humans and other organisms out there have more than just one gene. They have many. When we put all of these genes together, we call it an organism's genome. This includes all of the genetic information that is found within almost every cell in your body. Now, we have all this information figured out for humans, which was no easy feat. A group of scientists from all over the world established what was known as the Human Genome Project, which was completed in 2003, in which they worked together to completely map out the human genome, which is how we know certain genes occupy certain loci. Scientists literally mapped out every sequence of every chromosome. With this information, we can apply new data sets to it to compare, which is why you can spit in a cup, send your DNA to the lab, and then log into a database that has your specific DNA coded. This can provide insights into your health as it can screen you for specific diseases. This is done commercially and also clinically for many people all over the world, myself included. The human genome is a unique one, as are the genomes of all organisms. Humans specifically have 46 chromosomes, which are made up by having 2 copies of 23 unique chromosomes. We get a copy of each chromosome from our parents, which is why we have 2 copies of chromosome number 1, 2 copies of chromosome 2, and so on. Our 23rd pair of chromosomes we call our sex chromosomes because they determine the sex organs that are created while our body develops. XX for female and XY for male. What's unique about this chromosome map is that no other species on the planet has it. Only humans have 46 chromosomes that are put together this way. A dog, for example, has a total of 78 chromosomes, and a pea plant has a total of 14. Because the chromosomes of each species are unique, we can use chromosome numbers to help identify species and group them accordingly.